In this section, we are going to be looking at some typical organic chemistry reactions. We are going to be looking at nucleophilic substitutions, and this is an example of a substitution. You have a compound in which you have a hydroxyl group. The reaction happens, and in the final structure, the hydroxyl group has gone, and you have a new functional group, in this case, a chlorine atom. Addition reactions. Those typically happen to double and triple bonds. So you've got here cyclohexene, one double bond, and the final product is cyclohexane with 2 bromine added. You can also have eliminations, and typically elimination reactions will produce double bonds. So for example, here you have a hydroxyl group in your structure, and here you would have 2 hydrogens, and you end up and let's draw the other hydrogen from this carbon, and you eliminate from your molecule, in this case, well, an hydroxyl and a hydrogen, you form a molecule of water, and you form that elimination produces a double bond in your structure. And finally, you can have what we call rearrangements. So in this example, on one hand, you have an elimination. The hydroxyl group has disappeared and a double bond has appeared, but there also has been an arrangement. If you look at this structure here, in this carbon, you have 3 methyl groups, and you have 1 methyl group here, and there will be a hydrogen in this position. And if you compare that with the product, the final product, in the final product, one of the methyl groups has migrated from one carbon to the next. We will not look at these reactions this year, but you will see plenty of these examples of rearrangement reactions when we do inorganic and organic chemistry in your second year. Before we move on fully to study types of organic reactions, we need to be familiar with some key concepts. What is a nucleophile, what is an electrophile, and what's a leaving group? A nucleophile comes from, the word comes from the Greek nucleus loving, so loving positive charges. It's a negatively charged compound, either because it has a negative charge, so it's an anion, or because it has non-bonding electron pairs in the structure. So it behaves like a Lewis base. It can donate charge, it can donate electrons. Electrophile, it loves electrons. It's a neutral substance, or it can be positively charged, and it acts as a Lewis acid. So it has empty orbitals, which are low enough in energy, that are able of accepting electron pairs. So the nucleophile donates electrons, the electrophile accepts the electrons. Electronegativity plays a huge role in organic reactions because it determines the strength of a particular bond between carbon and that other atom, that heteroatom. So we've got here the example of the different bonds the carbon can make with a halogen group, and the strength of the bonds depending on the size of the atom and the electronegativity of the atom. So you have from very strong bonds, like the one between carbon and fluorine, to the weakest bond, which is the one that carbon forms with iodine. And on the other hand, you have the polarity of the bond. So when you have fluorine, which is a very strongly electronegative bond, it's the most electronegative. It's a very electronegative atom. It's the most electronegative atom of the whole periodic table. You have a high polarization of this bond between the carbon and the fluorine. There is a strong partial negative charge in the fluorine and a strong partial positive charge in the carbon. And if you compare that with chlorine, which is slightly less electronegative, and bromine, which is a even less electronegative halogen, and finally iodine, which is at the bottom of the group and is even less electronegative halogen, you move from a very, very polar bond, in the case of the carbon fluorine, to an almost non-polar bond in the case of carbon and iodine. It is difficult for carbon to stabilize positive or negative charges. It's difficult for carbon to act as a cation or an anion, but it can be done depending on the group that carbon is attached to. Sometimes you will have a carbon attached to a heteroatom, and the heteroatom will have a strongly partial negative charge. So when the bond between carbon and the heteroatom breaks, the heteroatom walks away with the electrons from the bond, and you form a carbocation. In other cases, the separation of charges is the opposite. Carbon is more electronegative than the heteroatom, which is this example here. When the carbon heteroatom breaks, is the carbon that walks away with electrons from the bond, and the heteroatom, the one that stabilizes the positive charge. So if the heteroatom walks away with electrons, then you form a carbocation. If it's the carbon that walks away with electrons that before were part of that bond, then it's how you form a carbanion. Carbocations act as a Lewis acid. They have a positive charge. They are highly reactive. They behave as electrophiles. They need electrons to complete the electronic needs, the bonding needs of carbon. So they react easily with a substance that is able to donate electrons with a Lewis base. And the reaction is fast, and carbocations in general are highly unstable, and they are formed many times in situ and then react, in the moment you need them, you create them, and they react quickly with any Lewis base in the medium. Having said that, carbocations are very unstable, and they react very quickly, and they often are created in situ right in the reaction before they are needed. Sometimes you can have very stable carbocations, and crystal violet, which is it's a dye, it's a purple dye widely used in biology, for example, for gram staining bacteria and also in the textile industry, is an example of a very stable carbocation. In this case, I've shown you, the structure is showing you the positive charge is sitting one of the nitrogens, but it is possible to have resonance structures in which the positive charge moves across the double bonds and the nitrogens from the structure. So you can have many resonance forms, many ways of stabilizing that positive charge by spreading it across the whole structure because of that ability of electrons to migrate. We call that resonance delocalization. That means the positive charge is not sitting in a particular atom, it is sitting across the whole molecule, and it can move around. And you have, on one hand, the conjugation of those alternated double bonds, and also the presence of three amino groups, three nitrogens with an non-bonding electronic pair that again donate electronic charge to stabilize that positive charge. In the previous slides, I discussed how carbocations were electrophiles. They had a positive charge, they were lacking electrons, they were attracted to substances that had electrons to donate. Carbon ions are nucleophiles. They have the electrons ready for the bond, and they act as Lewis bases. They have electrons that can donate and will react with Lewis acids to form the bond. Again, carbon stabilizes badly, negative charge stabilizes badly both positive and negative charges, so they tend to be very reactive species, and when they find another Lewis acid in the medium, they will be able to form a bond. And if you here, for example, you have the example of a carbon ion that will react with an acid. Imagine this is hydrochloric acid or bromidric acid, and it will attack the section of the acid that has the partial positive charge, and it will break the bond between the hydrogen and the heteroatom, let's say a chlorine or a bromine. And you end up with a stable organic compound and the counteranion of the acid leaving the molecule. And we call that section of the molecule that leaves, that is eliminated, the leaving group.